Imagine a vast library containing all the blueprints of life. This library is the genome, and the blueprints are our genes. But genes alone don't tell the whole story. They hold the instructions for building proteins, the true workhorses of our cells. Proteomics is the study of these proteins, their types, functions, interactions, and modifications. Proteins are the key players in virtually every biological process. They act as enzymes, hormones, antibodies, and structural components carrying out the instructions encoded by our DNA. Understanding the intricate world of proteins is crucial for unraveling the complexities of life and disease. Proteomics goes beyond simply identifying proteins. It delves into their abundance, modifications, interactions, and locations within cells and tissues. This comprehensive analysis provides a dynamic picture of cellular activity and how it changes in response to various factors. By studying the proteome, scientists can gain deep insights into the mechanisms of health and disease. This knowledge paves the way for developing novel diagnostic tools, personalized therapies, and targeted approaches to improve human health. Proteomics aims to answer fundamental questions about proteins and their roles in biological systems. One of its primary goals is protein identification. This involves determining the specific proteins present in a cell, tissue, or organism at a given time. Another crucial goal is protein quantification, which measures the abundance of each protein. Knowing the protein levels helps us understand their relative importance and how their expression changes under different conditions. Proteins rarely act alone. They interact with other proteins and molecules to form intricate networks that carry out complex cellular functions. Protein-protein interaction studies aim to map these interactions, revealing the intricate web of relationships within the proteome. Proteins undergo various modifications after they are synthesized, such as phosphorylation or glycosylation. These modifications can dramatically alter their activity, localization, and interactions. Analyzing post-translational modifications is essential for understanding the dynamic regulation of protein function. Mass spectrometry has revolutionized proteomics by providing a powerful tool to identify, characterize, and quantify proteins in complex biological samples. At its core, mass spectrometry measures the mass-to-charge ratio of ions, which are electrically charged atoms or molecules. In proteomics, proteins are first extracted from the sample and broken down into smaller peptides. These peptides are then ionized and separated based on their mass-to-charge ratio in the mass spectrometer. The resulting mass spectrum provides a unique fingerprint of the peptides present in the sample. The workflow of proteomics using mass spectrometry typically involves 5 main steps. Sample preparation, which involves extracting proteins from the biological sample and preparing them for analysis. Protein digestion, where proteins are digested into smaller peptides using enzymes like trypsin. Peptide separation, where the peptides are separated based on their physical and chemical properties using techniques like liquid chromatography. Mass analysis, where the separated peptides are introduced into the mass spectrometer, ionized, and their mass-to-charge ratio is measured. And finally, data analysis, where the mass spectra are analyzed using sophisticated software to identify and quantify the proteins present in the sample. Proteomics has emerged as a transformative tool in various research fields, including disease understanding, biomarker discovery, and drug development. By comparing the proteomes of healthy and diseased tissues, scientists can identify proteins that are differentially expressed in disease, providing valuable insights into disease mechanisms and potential therapeutic targets. In the realm of medicine, proteomics holds immense promise for improving diagnosis, prognosis, and treatment of diseases. By analyzing patient samples such as blood or tissue biopsies, proteomics can help identify disease-specific biomarkers, predict disease progression, and monitor treatment response. The development of personalized medicine is another exciting frontier in proteomics. By analyzing an individual's proteome, physicians can tailor treatment strategies to the patient's specific molecular profile, maximizing efficacy and minimizing adverse effects. Real-world examples highlight the vast potential of proteomics. Cancer proteomics, identifying tumor-specific proteins for early diagnosis, personalized therapy selection, and monitoring treatment response. COVID-19 research, understanding the virus-host interactions, identifying potential drug targets, and developing diagnostic tests. Agriculture, developing disease-resistant crops and improving crop yield. Pharmaceuticals, accelerating drug discovery by identifying novel drug targets and developing more effective therapeutics. In conclusion, proteomics is revolutionizing our understanding of the molecular basis of life and disease. By deciphering the intricate language of proteins, we are unlocking new avenues for improving human health and addressing global challenges. As technology continues to advance, proteomics will undoubtedly play an increasingly pivotal role in shaping the future of medicine and beyond.